Hi, today we'll discuss about tryptophan operon concept. So under gene regulation in prokaryotes, we have to study lac operon and tryptophan operon. Tryptophan operon. So the first question comes to your mind, what is tryptophan? Tryptophan is an essential amino acid. So amino acid means amino acids are required for the synthesis of proteins. And proteins are required for cellular activities. So what is tryptophan? It is an essential amino acid. It is required for the synthesis of protein, and protein is required for metabolic activities of bacterial cell. So bacteria requires tryptophan for its cellular activities. If bacteria requires tryptophan, bacteria has to synthesize tryptophan. You know two concepts under metabolism. There is a catabolism, anabolism. Synthesis comes under anabolism, breakdown comes under catabolism. We have studied lac operon. There, breakdown of lactose will occur. That's why it comes under catabolic mode. So lac operon is a catabolic activity. But now we need to synthesize tryptophan. That's why tryptophan operon comes under anabolic concept. So why we need to study tryptophan operon? Tryptophan is an amino acid required for bacteria, and we already know the operon concept, cluster of genes which controls the expression of genes. What is operon? We already know. An operon is a cluster of genes. For the synthesis of tryptophan, there are 5 structural genes. You can remember A, B, C, D, E, Trip E, Trip D, Trip C, Trip B, Trip A. Trip E codes for the gene required for the synthesis of anthranilate synthetase subunit. Trip D codes for the gene required for the synthesis of anthranilate synthetase subunit. And Trip C codes for the gene required for the synthesis of anthranilate isomerase or glycerol phosphate synthetase. The last two, that is Trip B and Trip A, codes for the gene required for the synthesis of tryptophan synthetase beta and alpha subunit. So all these 5 structural genes are required for the synthesis of tryptophan. So now we understood both the words, tryptophan, operon. Tryptophan amino acid and operon, 5 structural genes required for the synthesis of tryptophan. Now we'll study the structure of tryptophan operon. We already know. So operon, that is 5 structural genes, then operator, promoter. But if you observe the diagram, there is one more sequence called leader sequence. This plays a major role in tryptophan operon concept. So the structure of tryptophan operon includes 5 structural genes, leader sequence, operator, promoter. And one more additional thing is repressor. Repressor codes for the gene required for the synthesis of repressor protein. It is also called tryptophan repressor. In lac operon concept, you have studied I, that is regulatory gene. In the same way, here we are studying repressor. But repressor cannot directly bind to operator. That's why it is also called aporepressor. Now we need to study on and off mechanism. E. coli requires tryptophan, so E. coli has to synthesize tryptophan. When there is no tryptophan, E. coli has to synthesize. Therefore, the operon system must be on. See, there is a repressor and have mentioned this repressor cannot bind to operator. If repressor cannot bind to operator, transcription of all the structural genes will occur and synthesis of tryptophan will take place. But when there is high concentration of tryptophan, E. coli doesn't require to synthesize. When there is high concentration, obviously cell doesn't require again the steps, that is anabolic pathway. So operon system will be off. Operon system will be on when there is no tryptophan. When E. coli has to synthesize tryptophan, then on mechanism. When there is high concentration, no need of synthesis, therefore off mechanism. In the next video, I'll upload both on and off mechanism. Thank you.